Good morning, everybody. Myself, Dr. Shama from Swami Shri Swarupanand Saraswati Mahavidyalaya, Utruvilai. Today, I'll be taking your topic, Lytic development cycle of T4 Bacteriophage. It will be dealt under introduction, structure, types of life cycle, Lytic cycle, its significance, and the references which I have used. Introduction. What is a bacteriophage or T4 bacteriophage? They are the T4 bacteriophages are the viruses that eat bacteria, made up of bacteria plus phagine. Phagine means to devour. That means to eat something very fastly. It belongs to the T4 bacteriophage belongs to the order Cordovirales and the family Myoviridae. They were first reported by E.H. Hankin in 1896 that water of Ganges had antibacterial action against cholera. In 1915, Todd confirmed the presence of bacteria-eating viruses. These bacteriophages are non-enveloped. They have a contractile tail and double-stranded linear DNA with a protein capsid having 36 different types of proteins in its capsomeres, and they infect the colon Bacillus E. coli. T4 life cycle, the types of life cycle which this T4 bacteriophage uses to reproduce itself. There are two types of the life cycles which it uses, that is the lytic cycle and the lysogenic cycle. The lytic cycle, also known as the virulent life cycle, and the lysogenic is also known as the temperate life cycle. The major difference between the lytic and the lysogenic cycle are that in the lytic cycle, the viral DNA is free inside the host cell, whereas in the lysogenic cycle, the viral DNA gets attached to the host DNA. In lytic cycle, as the name suggests, lytic comes from the lysis. It involves the lysis of the host cell, and the number of copies of the virions are released for attacking the other host cells. And in the lysogenic cycle, the viral DNA gets attached to the bacterial DNA, reproduces along with the, replicates along with the bacterial DNA, and when it gets attached, that type of phage is known as lysogen or aprophage. Today, we will be discussing in detail about the lytic cycle or the virulent life cycle of the T4 bacteriophage. The lytic life development cycle of T4 phage, it says that the virus infects a host cell or a bacteria. Inside that, stops the machinery of the host cell and manufacture a large number of viral genomes and its capsids, then releases the virions and kills the host cell. The total lytic development cycle completes itself in 6 steps, which are the attachment, the penetration, the transcription, the biosynthesis, the maturation, and the release. What does we mean by the attachment? Attachment means the attachment of the viral cell over the host cell. Second is the penetration. Virus does not has its own machinery. It uses the machinery of the host cell, and for that it penetrates its genetic material inside the host cell and leaves the capsid on the outer surface. Third one is the transcription. In transcription, DNA is transcribed and translated into various proteins, and then the biosynthesis takes place, which includes the synthesis of replication of DNA and the protein coats, and the capsomeres are being synthesized. Finally, the maturation, that means the assembly, whole assembly is being arranged. The viral assembly is being arranged, head, tail, and is ready for the release. As soon as it matures, the cell lyses with the help of enzyme and the virions are free. The total process takes place within the 20 to 22 minutes, which we will be discussing in detail by the following diagram. So the first step, or the step 1, is the attachment. In this, the virus gets attached over the bacterial DNA with the help of specific receptors on the cell wall of the bacteria, and some chemical attraction also takes place. In as soon as it gets attached, the tail contracts and creates a tiny hole from which the DNA will pass. This leads to the second step, which is the penetration step. In the penetration step, as soon as the viral cell attaches on the bacterial cell wall, the phage DNA is injected into the bacterial cell by the contraction process, and the phage coat remains outside the cell. As soon as the viral DNA enters the host cell, the first and foremost work it does, it starts its own transcription. It starts the transcription and translation machinery using the machinery of the host cell. The host cell does not produce its own proteins, whereas the phage DNA transcribe producing mRNA and translates it into the phage proteins. A number of phage proteins are produced which are required for the replication of DNA of virus. In the meanwhile, the host DNA gets degraded by the enzymes and the proteins produced by the phage DNA. Replication of DNA starts, phage coat proteins are being made, other protein components are produced, and all the ingredients which are required for the assembling of phage DNA, phage gets completed in the biosynthesis phase, or the biosynthesis phase of the, that is the step 4th phase. Next is when all the parts of the host viral phage is being made, is the assembly part. The step 5th is the assembly, in which the phage components assembled. Phage components assembled means the head is being formed. DNA gets inserted into the head. It covers with its protein capsomeres. Tail gets attached. The tail fibers are attached, and the production of late protein, that is the lysozyme, takes place during this step. As soon as the lysozyme or the late proteins are synthesized or are produced, the in the meanwhile the virions get assembled. The assembled and mature virions are ready to be out from the host cell. The lysozyme present or the lysozyme produced by the virion viral DNA lyses the cell, and bacterial cell or the host cell, and army of virions are released, which is ready to attack the other host cells. In this way, in a short span of 20 to 22 minutes, we can the phage produces a numerous copies of phage virions. Now we will be seeing the time taken in the each process, that how these 20 minutes are being done. So as soon as it gets attached, the viral DNA starts to penetrate, as that time is being taken as the 0th minute, and when the DNA enters the host cell, it starts synthesizing, transcribing transcription to mRNA. It takes in a second minute. In the 3rd minute, the host DNA gets degenerated, and in the 5th minute, phage DNA gets replicated, and the late DNA RNA is made in the 9th minute, which includes various type of proteins which are and enzymes which are used for the assembly, for the protein coat and the assembly. In the 12th minute, head and tails are being made, which are being made with the help of the late RNA proteins in translation. In 13th minute, the heads are filled with the DNA, phage DNA, which is being replicated. In the 15th minute, the virions are formed, or the virions get assembled, head, tail, and tail fibers. In the 22nd minute, 15 to 22nd minutes, the lysozyme enzyme are being secreted, and it takes about 5 to 7 minutes to lyse the bacterial cell wall, and the release of virion take place. What is the significance of this Lytic cycle? Like living beings, viruses also live to reproduce, and they have to reproduce themselves. They find a host cell, replicate themselves, form their new copies, and fleeing a cell is the Lytic cycle. In the Lytic cycle, the virus reproduces thousands of times in 20 minutes, then weakens the cell wall, the cell lyses or bursts open, setting the army of new viruses free and result in the death of the infected cell. They alter the genome of non-virulent bacterial strain, producing more virulent strain. The references from which this topic is being dealt are the General Microbiology by Pawar and Ginawala, Microbiology by Dubey and Maheshwari, Microbiology by Prescott and Dunn. Thank you very much for listening to me, and if you have any doubt, you can contact me at my mobile number 997747166. It is also my WhatsApp number, or email your query at shamabaig@gmail.com. Thank you very much.